Okay, good evening everyone and welcome to the Lucy 50th monthly lecture series, a celebration of the 50th anniversary of the discovery of the iconic fossil Lucy by IHO founding director Don Johanson in Hadar on November 24th, 1974. I'm Chris Camposano. I'm an associate professor and research scientist here at the Institute of Human Origins, and I'm filling in for Johannes Haile Selassie, who is unfortunately out of town for the evening. First, I'd like to thank everyone who's here tonight, the IHO staff and the student workers for their organization and effort to make this lecture series happen. Also, I know we're wrapping up finals, so those of you that did make it, thank you very much. I'd like to thank the members of the IHO Executive Board who are supporting the entire year of the Lucy 50th celebration, including those who are here tonight. Thank you. And finally, to the IHO faculty who will be doing these talks over the next year, including myself in the fall. Thank you so much for helping to support one of the pillars of IHO's stool, or legs of IHO's stool, the public education and outreach about human origins science, especially in the important Lucy 50, a year of discovery. So as you guys know, we have a full year of themed monthly lectures, so please take a look at the schedule online and continue to attend these master classes in human origin science. Our in-person lectures will continue in September, and over the summer we have a few new lectures posted on IHO's YouTube channel. And with that, we'll move on to our speakers tonight. So first up, I believe, is Dr. Ann Stone, who is a biological anthropologist and Regents Professor in the School of Human Evolution and Social Change here at Arizona State University, also a research scientist in the Institute of Human Origin. Ann's research focuses on the analysis of genetic data to investigate population history and adaptation in humans, other primates, and pathogens. She is particularly interested in adaptation in the context of disease and diet, both of which have been particularly important over the course of human evolution. She is a fellow of the American Association for the Advancement of Science and the National Academy of Sciences. And with that, I will pass this off on to Dr. Ann Stone.
Thank you very much. It's a pleasure to be here and help celebrate this 50th anniversary. So, like most people, I love to hear great stories, and one of the greatest stories really is our origin story. And this is a story that can be told in many different ways, and also from many different perspectives. And so today I want to tell you a tiny sliver of that story that focuses on how ancient DNA tells us about how we have adapted over to our environment as it changes over our evolutionary history. But first I want to tell you a little bit about how we read the DNA to infer that adaptation has happened. And in genetics we talk a lot about the four forces of evolution. Mutation, which creates new variation, gene flow or migration, which moves it around, and genetic drift, which is sort of random chance over time. And then finally, what I'll focus on tonight, of course, is natural selection. Now, natural selection is something that we typically picture in thinking about positive selection. But generally, natural selection is really the differential contribution of some individuals to the next generation based on genetic variation and their ability to adapt. And so we talk about fitness, so in other words, not how buff they are, but really how many offspring, how many grandchildren, how many great-grandchildren. And of course, for there to be natural selection, there has to be diversity for it to work with. So you have to have variation existing in the population so that there are these sort of different choices, as it were. And over time, then, allele frequencies can change at one or many genes that influence a trait. And so this is how we kind of typically picture natural selection, but there are actually many different kinds of selection that are under the natural selection umbrella. So positive selection I just showed you, where one variant increases, but we can also have purifying selection, for example, where a deleterious variant is removed from the population. At some loci, we may actually have what's called balancing selection. And this is where you're really trying to maintain variation. So if you think about immune loci that are responding to all these different pathogens, having a lot of variation is often adaptive in those contexts. And so when you look over time, the amount of variation may not change that much, although the actual number of alleles, the exact types of alleles could change. And there are other types of selection that we might talk about. So, but how do we infer that adaptation has happened? So I want to kind of run through an example here to just give you a sense of how do we detect variation, how do we detect selection given the variation we see in the genome? And so here in this sort of image, what you see on the left is an example of a part of the genome where there's no selection happening. It's just sort of neutral. Each line represents a chromosome, and the little dots are variants, single nucleotide polymorphisms, okay? So changes in the DNA. You'll also notice that I've shaded the different colors, and these just demarcate different regions along the chromosome, sort of I binned them. And then within each bin, I can count the number of variants. And so these numbers, as we go through the generations, are going to change in some ways that'll help you see how we infer that selection has happened at some point in our genome. So here we have a new variant. I've put one on the neutral side and one on the positive side. The neutral one is just going to sort of sit there, but, we have to recount our number of variants, so the numbers have changed. But then as we go on through time, what you'll notice is that the positive side, the amount of variation will actually decrease. So 20 generations is about 500 years. When you move along to 80 generations, that's about 2,000 years. Our allele frequency of the variant that's being selected for increases, but the general level of variation in that region has really decreased, okay? So whenever we're scanning the genome and we see a region that suddenly it has much less variation compared to the rest of the genome, that's sort of an alert that something might be going on there, okay? There's a lot of different tests of selection. I'm not going to get into the details of that take advantage of different aspects of the data statistically and think about that. But this is basically how we read genomes to figure out, is a particular region important? Now, that's a modern genome, but how does ancient DNA data help? And it helps in several different ways. One, we actually have the ability to look into time, at different slices of time, to see when did this variant start to increase, for example. We can look at the pace or pattern. Does it start out, just sort of increasing slowly or rapidly? And is this true over a geographic region in different populations or just in a very limited region? We can try to make inferences maybe about the origin of the selective variant. So we could maybe see that there was a migration into a region where, in the past, where the migrants introduced a new variant that then was subject to selection. And if we're looking at these different slices through time, we can also kind of think about whether there are changes in selective pressure over that time. So has the climate changed, for example? If it's become drier, you might expect adaptations to aridity, for example. Or if you see evidence for a particular disease being present, that might signify change. So the first example that I want to give, I'm really going to focus on these first two, timing and the pace and pattern of selection. And the example I want to talk about is lactase persistence. And so lactase persistence is the ability to digest milk into adulthood. It's evolved independently in several places around the world. But the example I'm going to give you today is from Europe, because we have the most ancient genome data from Europe. And so we have these really lovely time slices that we can look through to see how the allele frequencies for lactase persistence have changed, or how they've, what the allele frequencies are at different time points. Now, it might not surprise you, or one hypothesis might be that this was selected for as agriculture is introduced, right? And particularly dairying. Now, in Europe, agriculture, depending on where you are, arrives about 7,500 to 4,500 years ago, right? It's later in the north, earlier, closer to the Middle East. What you'll notice here, though, is that there's kind of a lag between where you start to see the alleles for lactase persistence to actually start increasing in frequency. And you'll notice that the modern levels are quite a bit higher even than it was 1,000 years ago. You'll also notice that we don't have equal amounts of data from throughout the region as well. So that's another thing that will be interesting to look at as time goes by. But this lag actually opens up some really interesting anthropological questions. So what role did cultural adaptations play? If you know that people are dairying, how are they using the milk that is produced? One way to break down milk sugars, lactose, is to use bacteria. So to eat yogurt, to make cheese. And we can look in the archaeological record and see that people indeed did that. Our microbiomes also can help digest lactose. And so populations in Mongolia, for example, who eat a lot of dairy products, have a very healthy microbiome that kind of does that for them. So that's one thing we can think about. We can also ask, were there migrations that introduced this into the region, and does that shift things at a certain time? One migration during the Bronze Age is the Yamnaya, and we can see from their genomes that they did not have the lactase persistent allele. So this basically tells us where the origin wasn't in that case. My second example is looking at this question of origin. Are there cases where we can discern where the variant that was selected came from? And we have a wonderful example when we look at high altitude adaptation. Now, high altitude adaptation is also something that's happened independently in multiple places around the world. Here I've shown the lowlands of Peru and the highlands of Peru. Also occurred in Ethiopia, in the Simian Plateau, in highland Papua New Guinea, and in the Tibetan Plateau, which is what I'm going to talk about. So what you tend to see is lowland populations where maybe allele frequencies don't change compared to a set of loci, and these are different in different parts of the world related to adaptation to high altitude. So Amelia Huerta Sanchez and her collaborators focused on Tibetan plateau populations, and in comparisons of different parts of the genome among populations, they were looking for, regions that really stood out as being different, right? There's some variant that makes that population just at that part of the genome look really different from everybody else. And so one of the loci where they see that is EPOS1, and this is a gene that's involved in the hypoxia pathway, so adapting to low oxygen which you, of course, find at high altitude. Now, when she and her collaborators went to sequence the region around this gene, they found a very interesting pattern. And so here the derived alleles are in black and the ancestral alleles are in white. And you can kind of see by eye that there's a very different pattern in Tibetans compared to lowland Han Chinese. You'll also see that right in the EPAS1 gene, there's kind of this blue highlighted section which seem to be very important and have functional implications. And one of the things they did was take all the alleles at this gene and connect them through a network showing the relationships. And I want to point out a couple of things. Here's the Tibetan allele. It's kind of off on its own. It's actually 40 changes different from the nearest other population. The Han Chinese are sort of in yellow, scattered throughout the network, and not particularly, their allele is not particularly closely related to the Tibetan allele. The Tibetan allele, however, is very closely related to an allele we find in archaic hominins, and specifically in the Denisovans. The Denisovans are known basically from their DNA, from Denisova cave, and it's really intriguing that this allele appears to have been given to us from Denisovans into modern humans, and then at some point became quite adaptive in the context of high altitude. And I think one of the things that ancient DNA has been, that's been amazing and super interesting over the last 10, 15 years is the information that we've retrieved from archaic genomes in comparison with modern humans. And from this article, what you'll notice is we know that archaic and anatomically modern humans admixed, interbred, and we can look through the generations at which DNA is still present in human populations today and which DNA we don't see. And so the example I just gave you is really a classic signature of adaptive integration that was adaptive in the high altitude context. And there are others. But there's also another, you can sort of reverse this and ask, what do we not see, right? Which parts of the human genome today do we never see an archaic variant, right? Because maybe that tells us something about how anatomically modern humans are different from archaic humans. And that could have been purified. This would be an example of purifying selection from our genome. So we do have an example of this. Trujillo et al. Asked the question of where do we see non-synonymous changes, so changes that change the protein, where humans are completely fixed for a derived variant and the archaic humans are fixed for the ancestral variant. And so they looked through the genomes of modern humans. You can see an example here. And then when they looked closely, this was in a gene called NOVA1, and specifically in an RNA binding domain. So this gene, actually what they did was super cool. They used genome editing to introduce the archaic version into pluripotent stem cells. And then they grew little cortical organoids, so like these little kind of tissue brains in a petri dish, but tiny. They only grow a little bit. And so they differentiated the pluripotent stem cells into these little organoids. And what they noticed was this change caused kind of changes in a bunch of genes in how they spliced and interacted with other genes, and particularly genes involved in neurodevelopment, in proliferation and synaptic connectivity, right? So something important in how our brain develops in modern humans. Finally, I want to talk about some examples that tell us about how selective pressures have changed and how we've responded. And a classic example, of course, is disease. And one that we all know about is the Black Death. So at the top, what you can see is how Klunk et al. Have used ancient genomes before, during, and after the Black Death to ask what were the selective pressures, what genes were more frequent after the Black Death, what alleles, I should say, and if there's some alleles that help protect you, are there other alleles at that same gene that give you more susceptibility, right, to dying? And so they did this, and you can see 4 loci that are highlighted in yellow that were found both in London and in Denmark in populations before, during, and after as being important, as having significant shifts in allele frequencies. Then the other thing you can ask is, focusing in on that was just the comparison to identify these. How do we see these allele frequency changes? And you can see the London in red before the Black Death, during, and after these specific variants. So this is one that increased, the first one increased in allele frequency, but you'll notice that those who died during the Black Death were less likely to have that version. The last one is the opposite trend. And if you think about how pathogen loads change over time, this might have been advantageous some of these might have been advantageous to help you survive the Black Death, but maybe disadvantageous for some other pathogen. And we know that some of these alleles actually are known risk factors for Crohn's disease, for example. One final example is in South America, but instead of Black Death, we're talking about tuberculosis. And this is a modern genome scan of South American populations, and it highlighted quite a few loci that seem to be important for resistance in tuberculosis. And they hypothesized that these variants that confer protection against TB started increasing in frequency about 120 generations ago, about 3,000 years ago. And my own lab, we work on the evolutionary history of tuberculosis complex bacteria, and Mycobacterium pinnipedi appears to, from ancient tuberculosis genomes, we can infer that it arrives in South America about perhaps 2,600, 2,500 years ago. And so this seems to fit nicely. And with my collaborators, we're now looking on the human side to see, do we start seeing these changes in allele frequency as time goes by? Now tonight we're celebrating the 50th anniversary of the discovery of Lucy, and so you might ask, can ancient DNA help us understand the evolutionary pressures that she faced and her people faced? And unfortunately, I think the answer is perhaps no. It's very difficult to use the genome to reach that far back in time. And unfortunately, she did not die in the middle of a snowdrift and get buried in permafrost in Siberia, because we do have cases, at least, where we get DNA that's over 1 million years old. But there are new technologies that are allowing us to get information from proteomic data, so from protein data, and at least ask some questions about relationships among Australopithecines, for example, in Africa more than 1 million years ago. So hopefully that will continue to progress. Finally, I'd like to thank my lab and the funding agencies over the year. So thank you very much.